I'll go over our Western blot analysis, which was more specific for each protein that we indicated with the mass spectrometry. So first, to model our ethanol drinking, we had male rats, and they were put under experimentation for 4 weeks. The first group I'll refer to either as the continuous group, continuous ethanol, or the CE group. And basically, they were given access to ethanol for 24 hours a day over the course of the entire 4 weeks. The next schedule is called, I'll either refer to it as a multiple schedule, the MSA, or a binge-like access. So basically, we wanted to model binge-like drinking. So these rats were given ethanol for 1 hour a day, 1 hour at a time for 3 times a day during their dark cycle. So because they were given ethanol over a short amount of time, that modeled a binge-like drinking. The last group was given water instead of ethanol, so we had a basis to model our results on. So here we have graphed the amount of ethanol that was intaked by each group. So on the left, we compared the MSA and the continuous group. For each of the weeks, the continuous group did have a higher intake than the MSA group, but the only significant difference was in week 1. On the right, we have the amount of ethanol that was consumed by the rats only in the MSA group. So it goes on based on how much ethanol they consume based on each hour. So with this data, we were able to better analyze how their behavior affects the effect of ethanol. So after the 4 weeks, we dissected the amygdalas from the rat brains, and we used a homogenization process to process the protein samples for further analysis. So next, this is what my mentor did before I started my internship, but he conducted a mass spectrometry analysis. So basically, the protein samples were subjected to a reduction and a tryptic digestion process. Either a light, intermediate, or a heavy dimethyl labeling was performed on the peptides for each of the sample groups. The labeled samples were then desalted, and a software called the mascot distiller was used for identification and quantitation of the proteins. So basically, this method was able to scan a large amount of proteins for significant changes, which we then looked at more specifically with the Western blot. So this is the data that we collected from the mass spectrometry. On the left, we chose these proteins because they showed the most range of significance. These numbers are the ratio. So in the first column, we have the ratio between the MSA group and the water group based on the expression, how much expression that the protein showed. And the second group is based on the second column is the comparison between the continuous group and the water group. So these are just ratios between them. They're separated into 3 sections. The first section, the blue one, represents a significant change that occurred in both the MSA and the water groups. The pink group showed significant changes in only the MSA group, and lastly, the green group only showed significant changes in the continuous group. So this is the data that we wanted to confirm with the Western blot analysis. So with our Western blot analysis, we used the same protein samples, and we loaded them into each of these wells. And basically, we did a gel electrophoresis, which separated the protein based on weight. So next, since the gel is very sensitive, we needed to transfer it onto a membrane. So the proteins were electrophoretically transferred to a PVDF membrane. And this diagram shows the transfer sandwich. So the gel originally had all of the protein sample, would have been transferred onto this membrane for further analysis. And then after, we would take our membrane and cut it based on where we expected each protein to be located. And we blocked them in 5% milk and TBS to prevent nonspecific binding. We then incubated the blots in the corresponding primary antibodies at 4 degrees Celsius overnight. Incubating it in the primary antibody binds to the protein that's on the blot. After we did the primary antibody incubation, we did the secondary antibody incubation at room temperature, And so the secondary antibody binds to the primary antibody, and then we're able to detect the protein. So after that, the blots were incubated in a detection reagent, and with that, we were able to analyze it under the Kodak imaging machine. And after we received the image, we could quantify the intensity of each of the bands to quantify the amount of expression using an imaging analysis software. So these are the proteins that we looked at more specifically. Each of them have a different neuronal function that is important to the functioning of the brain. So for example, the first one, PSD95, has an important function in learning and is associated with autism. They vary from learning to neuronal diseases. So Dreibrin right here is associated with Alzheimer's disease. So all these proteins have an important function, so we wanted to analyze these specifically. So here's an example of the one of the proteins for the Western blot data we recorded. So this blot right here is what we collected. The one below here is coherin, and coherin is one of the housekeeping proteins that we normalized our protein of interest to. And this housekeeping protein is necessary in order to account for any human errors or basically to make sure that our data is consistent between each of the gels themselves. So these, each of these individual bands were quantitated based on their intensity. So this band would have more intensity than this band, which you can see is lighter. So with that, we quantify each of these and normalize it to this. So we receive the ratio between the two. The first four correspond to the binge group, the second four correspond to the continuous group, and the last four consist of the control group. So these 4, 4, and 5 were averaged and are graphed here. So the only significant difference that we found was between the continuous group and the control group, which does confirm our mass spectrometry data, which is listed right here. So here's additional data onto more proteins that we looked at. So on the left is EAT1 and 1433. They were both normalized to coherin as well. And so the only significant difference for this one was the binge group, and the only significant difference over here was in the continuous group. And it's more noticeable on the second slide. Yeah, so over here, the differences between the binge and the control and the continuous and the control are significantly different. So we can kind of assume that the ethanol affects the binge group and the continuous group differentially, since their levels are not exactly the same. Also, you can see on the blot over here, it becomes more faded, so that's why the difference is more significant in this blot. And lastly, we have Dreibrin and transgalolase, which these two were normalized to GAPDH instead because we had some difficulties with the stability of coherin. And again, the difference between them is noticeable, so binge and continuous show differential changes, and the binge and the continuous show differential changes as well. So we can conclude that ethanol drinking alters the expression of key proteins that are important for neuronal function. We can also say that the binge and the continuous drinking show differential effects on the protein expression in the amygdala. We were able to confirm our mass spectrometry results because because our data we collected from the Western blot was consistent with the mass spectrometry data that we collected, that my mentor had collected previously. And so these results are important in directing and better understanding the biochemical mechanisms of how ethanol affects the brain, but also it will help direct treatment towards more specific, a more specific target. And so in the future, we would like to investigate more proteins so we can better understand the overall effects. And also, we would like to increase the trials because many times the Western blots could have been a little inconsistent due to human error, so we would just like to repeat more trials just to confirm our data. And so I would like to thank my mentors, Dr. Bill Huang and Dr. Hyun Kim, for allowing me to work at their lab this summer and guiding me through all of the procedures. I would like to thank my SMACS teachers, Mr. Curran and Ms. Serene, for guiding me through SRP. And I would like to thank my family and friends for all of their support.
Can you go back to the slide with the list of the proteins? No. So I understand that your project was more about whether or not these proteins were affected by alcoholism or whatever. But do you know how deeply does it actually change the function, or does it just change the structure or whatever? Because sometimes if you change the structure, the shape can still remain similar and have its same function. So do you know how it affects the function?
We don't know how it affected the function specifically. We were just looking at how much the protein was expressed.
Oh, okay. Okay.
Any other questions? Yeah, Connor.
Okay, so I know that ethanol binds to certain neurotransmitters in neurons. So do you know whether that interaction is what causes the decrease in protein expression, or is it the toxicity of the ethanol that causes it?
I'm sure both affect it, but we mainly looked at just the expression, not how it exactly affects it. Yeah, Teresa.
What is your favorite part of it?
So I worked at the Twinbrook campus of NIH, and so I didn't have many people to eat lunch with who were my age. So whenever I went to Bethesda, I always ate lunch with Lydia Wang and Justin sometimes, and Connor.
Me?
So yeah, I saw you a couple times.
What? I didn't even work
for
there.
Poster day. Anyway,
When you came in too.
it was nice to see them all over the summer. Yeah, pretty.
Do you plan on testing this on, different animals?
Well, for our experiment, we chose, the rats that we used are called P rats. They're alcohol-preferring rats, so they're genetically modified to prefer alcohol. They're modeled to, they're modeled after humans, so they would, so that the results we receive from this would eventually apply to human studies as well. So we haven't gotten there yet. Yeah.
What was the most challenging aspect of it?
I think just being patient, because there's a lot of bleeding when it comes to Western blots, and repeating the same procedures over and over again took a lot of patience. So I think my internship taught me how to become more. Yeah.
What was the least favorite part about your internship?
All the waiting.
Okay.
Being patient.
Yeah.
Okay.
Any other questions?
So did I.
I'll guess.
Is that okay?
Yeah, it was.
You try to get her.
No.
You got that? You asked?
Oh yeah, I got the whole thing on video.